What grows in simulated lunar regolith? That's the question I'm going to answer today in Science Spotlight 15, Simulated Lunar Regolith for Plant Experiments. I'm Dr. Mark Williamson. Let's get started. So, we should define what regolith is before we get going here. So, regolith is unconsolidated material covering bedrock on a planetary body, and it includes things like dust, broken up rocks, even soil, and other related materials. At least soil when we talk about Earth concept. Because there's actually, when we talk about like lunar regolith, Martian regolith, regoliths from non-Earth planets, we're talking about something that is not soil. So regolith isn't synonymous with soil, even though sometimes in the literature they call it lunar soil. It's not really soil because soil contains organic matter and a microbiome. Regolith, at least extraterrestrial regolith, does not. Now, there's different kinds of regolith. The most notable ones are from the moon and Mars. And from the moon, there's lunar Mar, lunar highlands, so from the different major regions of the moon, as well as lunar dust even, and of course, Martian. Now, there are going to be different subtypes, but those are the big picture types when we talk about regolith. Now, true regolith is not easily available. We don't have any from Mars yet, and only very vanishingly small amounts from the Apollo missions from the moon. So most research uses what's known as simulants. So they simulate the properties of real regolith, but are made out of minerals, materials from Earth. There's different companies that will do these, different universities. One commercial source is Space Resource Technologies. In fact, that's where the pictures for these simulants come from, and so you can even buy them yourself. And now, when we talk about regolith research, there's a variety of keywords that come up over and over again, so it's good to get our heads around them. Similar lunar regolith, or SLR, so that's lunar regolith that is made from Earth materials, so it's not real, but it mimics real regolith. There's bioregenerative life support systems, or BLSSs. So if you think about a moon-based space station, any place where you're in space and you have to keep life going, you're probably going to be including biological systems like plants, bacteria, algae, etc. And so regolith, especially when we talk about like a moon base, a terrestrial planet base, regolith and growing plants with regolith are going to factor into these bioregenerative life support systems. Then there's more broadly the term of in situ resource utilization. That's like if you go on the moon, instead of having to ship everything there, you take advantage of the resources there, the resources in situ. And so regolith would be one of those resources. And then finally, regolith-based agriculture, of course, growing plants, growing crops using regolith. So a good review of regolith simulants for plant growth is from the 2022 article, The Potential for Lunar and Martian Regolith Simulants to Sustain Plant Growth, a multidisciplinary overview. So that's that paper here. And so the main point is that extraterrestrial human simulants can't rely on perpetual shipments of materials. They're going to have to use stuff there, so that in situ. And one of those resources are going to be is going to be regolith, especially for growing plants, a substrate for growing plants. And there's a lot of different types of experiments. See, the most obvious one would be plants, but you can also include animals in there, like worms, fungus, and even microbes. And plants have certain elemental needs, so there's going to be a contrast between what the actual regolith has and what plants actually need. So plants need carbon, nitrogen, phosphorus, potassium, sulfur, calcium, magnesium, and to a lesser degree, other elements, including heavy metals or just metals. And we'll see a little bit of the composition of regolith in a moment. And why you would grow plants on extraterrestrial settlements. Well, obviously food production, but also things like water recycling, organic waste composting, oxygen production, and CO2 consumption. So plants taking CO2 to emit oxygen, and even psychological comfort for the astronauts. Now, regolith is not easy to work with. It's not just grab and go. Most regolith is very alkaline, so it has high pH. There are toxic elements like aluminum and chromium and in precipitates, and even perchlorate salts on Mars. So lunar and Martian regolith will have different dangers. Particle size distribution can be a problem. Sodium levels, water holding capacity. Regolith does not hold water very well. It doesn't circulate air very well. And it's unknown exactly all the interactions with biota. So regolith might not be a great situation when we have to come up with pathogen reduction that we know fairly well about earth soils, about how pathogens interact, but it's less known about regolith. So the take-home point is you can't just use regolith straight to reliably grow plants. You're going to need to have some sort of improvement. Nutrient amendments to take care of the low nutrients, either organic or inorganic. Even doing things like pioneer plant growth, so growing plants that you don't care about like harvesting, but you want them to prepare the soil for the crops later, or even dealing with microbial or other microorganism consortium in the regolith to essentially go from regolith to something closer to a living soil. And so the different types of regolith will have different compositions, but they're pretty heavy in silicon, oxides, other materials, and we'll see in a little bit here, the biggest downside is some of the things that plants really need from the soil: organic carbon, nitrogen, to a lesser degree, potassium are not available, very low in the regolith. So thinking more about space agricultural and simulants themselves, because again, we don't have a lot of real regolith, so we need to simulate it. And that ties into this bigger picture of bioremediation. So bioremediation is using biological agents to transform infertile or contaminated substrates into a more life-supporting form. So regolith is by definition infertile. So you're going to need to bioremediate to actually use it. So by detoxifying harmful things and improving, so there's positive things like changing the chemical properties or adding elements that the plants need. And one thing to note that even if the mineral elements are present in regolith, they often have low bioavailability. The plants can't utilize it unless you make it bioavailable through microbial methods, etc. And there's been little research to characterize microbial communities that naturally occur in lunar regolith simulant. Now, regolith on the moon is totally sterile. There's no living things there, but of course, while it's not very hospitable of the simulant on Earth, there still could be microbes. And so any studies that deal with microbes that doesn't sort of characterize what's already could be there before you add things to it is going to be throwing things off. Big take-home point when it comes to simulants, when it comes to plant growth: nitrogen. Nitrogen, plants need a lot of nitrogen, and regolith has almost no nitrogen. So it will need to be supplied and recycled for sustainable space agriculture to even take place. Phosphorus, now it is present, but it needs to be made bioavailable. So that's less of a not there, but more of a unlocking it problem. And to generate simulants, there's over 40 that have been made by 13 different nations, including commercial offerings. Space research technology is one example that are already given here, but there's things like off-planet researches, labs that will produce it, NASA, etc. And so there's a lot going on. So it's a growing field of research. And though different simulants can't perfectly replicate everything from real lunar regolith, but some are better than others for certain purposes. So hopefully in the future, this is a potential project from the Colorado School of Mines, having this regolith simulant report card to give reports on the different types of simulant. And hopefully that will be a future potential resource that we can avail ourselves of. Anyways, so here's some nice tables here of sort of the primary nutrients, so nitrogen, phosphorus, potassium, the uptake form, what the plants actually require, what the actual lunar highland in Mars has, or sorry, lunar highland in Mar, and it's very low for nitrogen. Okay for phosphorus, but needs to be unlocked. And then potassium kind of depends on where you're at. And when you're growing plants, you're going to need to select different characteristics for your particular outcome. All right, so we've talked about regolith, a little bit about plant growth, but let's zoom out to the broader picture when we talk about regolith and research. So this is a overview of the lunar in situ research utilization techniques for future lunar missions. So the big shift is the next stage of lunar exploration is shifting from understanding the moon to actually making use of the moon. And so the idea of having a permanent moon base with the Artemis program, etc. So some focus areas. If we're going to have long-term habitation of the moon, you're going to need to supply life-supporting materials, water, oxygen, energy, and yes, food. But food, so growing plants in something like regolith, is only a part of the overall picture that we need to solve and where regolith may play a role. There's also construction and manufacturing of facilities, including creation of rocket propellant. Of course, there's going to be scientific research and transit and supply stations. So water, oxygen, other material extraction will need to take place in the regolith. And that lunar, quote unquote, soil could be used as a substrate for plant cultivation. So using regolith for plant cultivation is one part of the overall big picture. And as I've said before, direct use as a substrate is not sustainable. You have to remediate or supplement the regolith to make it useful for agriculture. And so there's some figures here talking the top one about the different types of soils or regolith more accurately and what might be done to them to extract the things needed. Lunar regolith has also been studied in actually 3D printers to create substrate, like building materials. And then some of the different concentrations of various volatiles in solar wind and implanting into the lunar regolith. All right, so let's turn to real regolith studies. This is probably some of the most fascinating studies. It's using actual regolith recovered from the moon in the Apollo missions. So a good review here is the Lunar Plant Biology, 2010. So we have regolith samples from the Apollo era. And when we first return them, they're more broader astrobiological questions like what's the limit of terrestrial biology and adaptive processes. But slowly we started to think about how the regolith might be used to grow plants. And it's really interesting because you see pictures and there's a lot of contamination protocols. They really sterilized everything and made sure there isn't a lot of contact before they knew more. Because actually one of the original worries bringing samples back to their moon is this term called back contamination, which is the idea that samples returned from the place like the moon from an extraterrestrial environment might harbor either alien organisms that are pathogenic or harmful, or substances that could cause mutation or pathogenesis. So it wasn't clear, like, is regolith like dangerous? Like, will it hurt plants and will it hurt people? And so they're very careful. And so they ran preliminary analysis and they confirmed what's pretty obvious now, but wasn't back then, is that there's no evidence of any previously living or fossil material in lunar regolith. And then they did some growth studies where they grew plants not in regolith. They actually grew them sort of in some substrate, some soil or something like that. But then they sort of dusted real regolith on it. And they didn't find any adverse effects. So the growth of plants in contact with lunar materials, not in lunar materials, but in contact, it was confirmed. But no actual experiments with regolith were done as the primary growth medium. However, that actually changed in 2022 with the article Plants Grown in Apollo Lunar Regolith Present Stress-Associated Transcriptomes That Inform Prospects for Lunar Exploration. They actually did it. There's a study, grew Arabidopsis, the model plant organism or model organism for the plants. They grew them and actually germinated them in Apollo 11, 12, 17 samples, and then they used this simulant as a control. So they had these trays, very little bit of regolith, and they put the plants in and they watered them and they got them to grow. They measured plant growth and then gene expression. And what they found is, yeah, they were able to get plants to grow. The ones in the real regolith were slower to develop, and many actually had severe stress morphologies. They actually had three different morphologies. They had the large lunar plants, so that kind of grew more or less normally or comparably to the simulant. There's the small ones that did get very big, but otherwise looked all right. And then there's the severe morphology, which were like really shriveled and almost dead. And with gene expression, they found differentially expressed genes indicating stresses on those plants, especially with salt, metal, and reactive oxygen species. So here's a bigger picture here. You can see the different types. You can see a bit of the site information for Apollo 11, 12, and 17 that you can go into more in this paper. All right, so given that, we have Apollo regolith, we now have simulants. Surely there's a bunch of NASA studies. Well, I looked at the Open Science Data Repository at NASA, and actually only found a handful of papers about lunar regolith. Well, one of them was the one we just covered here. There's a couple others we'll see later, and then some were actually just Martian. So not a whole lot of official studies, or at least studies that have been officially cataloged with data availability in NASA's Open Science Repository. So now we can turn to more review type articles for simulant studies. The first one here is 2024, How to Make Lunar Soil Suitable for Cultivation. And so there's still no well-developed system for lunar soil improvement. And so this review evaluated existing methods and research gaps. One important point is that algae and bacteria are significant for soil functionality. So it's not just the plants and the regolith, it's also the microbiome. And there's a lot of shortcomings of lunar regolith. We've covered some of these before: low air permeability, poor water and nutrient holding capacity, isn't really conducive to microbial growth. And in fact, the physical structure of the particles are actually sharp. They can actually damage plant roots. And there's, of course, an absence of soil organic matter. And one thing to note, true regolith might actually be more barren than typical simulated varieties. So it'll actually likely be harder using real regolith than the simulated stuff, unsurprisingly. And of course, we have to deal with gravity, etc., in situ on the moon or other planets. And so it's typical to actually, in these types of studies, to not just use pure regolith, to include additives up to 20 to 80% by volume. And pre-experimental sterilization, at least for standardization, is probably important for accuracy, though it's kind of hard because sterilizing regolith can change the chemical properties. So it's sort of a grab bag of kind of what you're trying to do and how it'll affect it. You'll have to consider that into an experimental design. Anyways, what this paper kind of came out with as an improved process, not necessarily for every experiment, for probably how it would work in a real like lunar situation. This is the process they came up with. You're going to want to start by sieving regolith to get suitable particle sizes, then mix it with fermented organic fertilizer. Here they suggest feces and straws to get the carbon and the nitrogen. Then sterilize the mix to protect from pathogens, then add back in beneficial microbes to enhance plants' growth, and then those other things that are needed, water, potassium, phosphorus, are going to need to be brought in through extraction and bio-weathering. And so, nice picture here of all the things that can go wrong and hurt the plant, from mechanical injury to high pH to nutrition and salt stress, etc. There's also a picture here or a schematic of the overall sort of bioregenerative system and how lunar soil need to be improved and factored into all the other life support type things. All right, another review here is overview and recommendations for research on plants and microbes and regolith-based agriculture. This is 2024. So these studies are in the infancy. They need more established parameters for standardization and validation. And this review recommended different tiers of like types of research: introductory research, intermediate, and advanced. I thought that was pretty helpful. Brought up that you're probably going to be doing this in parallel with hydroponics and soilless substrates. And as we've been talking about before, lack of nitrogen, phosphorus, and potassium is a significant challenge, both Mars and the moon, and then salinity is a problem on Mars. And one place on the moon called like the creep, lunar creep, it's the concentration places with the terrain that has relatively high concentrations of potassium, rare earth elements, and phosphorus. That would probably be the best place to try to get regolith for plant growth. There are relevant microbial contributions that need to be considered, like plants growth promoting bacteria and nitrogen fixing bacteria, which will likely be vital for actually creating a substrate that can sustain plant life. And there's a lot of critical knowledge gaps, from the effects of sterilization to challenges for DNA extraction and sequencing. If you're trying to sequence the soil microbiome that you develop through this regolith, and etc. You might actually want to consider, for standardization purposes, a more custom-built growth chamber that's a place to reduce a lot of these environmental variables when you're doing these studies. Anyway, so a lot of important tables. We've seen tables like this before, but it's always good to have more of this. The different plant nutrients and their compositions across the moon and Mars, the introductory, intermediate, and advanced designs and considerations for those, as well as different recommendations for measuring plant responses, starting with the introductory, intermediate, and advanced types of studies. And they have these decision trees for overall regolith experimental design, for plant experimental design, and even microbe experimental design. All right, so let's get out of the reviews into some notable specific studies. So let's start with plant studies, because plants be the most obvious thing to look at. So this is this great paper. I could only get the abstract, but this paper called Sporobolus areoides as a pioneer plant for lunar regolith. It really showcases a really, I think, important sort of stepwise process of probably not planting crops directly, but like planting pioneer plants to help turn the regolith into something closer than soil. And so that's what they did here. Here's just the abstract, but you can look at the bottom here where I pull out the important bits. So this Sporobolus, it's also known as alkali sacchaton. It's a hardy species of grass, and they actually grew it in regolith. They tried another species that didn't really work that well. And it actually grew as well in sand as Lunar Regolith simulant. So this is a picture of the grass. So it could be a potential pioneer plant to grow in Lunar Regolith. A more conventional study here back in 2014. This is actually the first large-scale controlled experiment to investigate growing plants in Mars and Moon soil simulants, or regolith. They actually had 6 different wild species of plant, 4 nitrogen fixture species, and 4 crops. So there's table 1 here. They had leopard's bane, field mustard, stinging nettle, marsh thistle, reflex stonecrop, and red fescue as the wild plants, common vetch, lupin, yellow sweet clover, and greater bird's foot trefoil as the nitrogen fixtures. And then the crops were tomato, rye, carrot, and garden cress. They grew them in small pots up to 100 grams of simulant, or sort of the soil control. They had 20 replicates per species, per soil types. They had a total of 840 pots. So this is just one picture of one of the sets of blocks. And so there's a lot. This is a big study. This is a really well designed study. And they did find that most of their species they grew reached germination. The Mars regolith simulant was overwhelmingly better at growing than the Lunar regolith simulant. I don't think they took into account the fact that Mars has these perchlorate salts, which are toxic. But at least if you could get those out, see Mars was easier to grow things than the lunar regolith. And one of the result sentences was reflect stone crop, a wild plant, the crops, tomato, wheat, and cress, and the green manure species, field mustard, performed particularly well. The latter three flowered, and cress and field mustard also produced seeds. So not only did they get plants to germinate, to grow, some flowered, some even produced seeds. So the next generation of plants. And oh, if you look at the left here, this is what I'm talking about. This is 20 blocks, each of the individual pots. The red is Mars, Martian, the yellow is lunar, and the green is, I believe, a Earth like analog. Or it was a like a deep, low nutrient, like river mud or something. So low in nutrients, but sort of the Earth negative control. And they just have all of their blocks there. It's just glorious from an experimental design procedure. Anyways, this is some of the results, the various metrics from germination to being alive at a certain time, biomass, etc. And you can see that there's differences across different plants. All right, let's shift over to microbial studies, notable ones. There's one in 2026, effects of microbial fertilizers on the properties of simulated lunar soil and lettuce growth. So while the previous one was a huge, large scale, this is much more modest. Pretty introductory, but nice little study. They tested 3 commercial microbial fertilizers on improving simulated lunar regolith and promoting lettuce growth. And they evaluated that. They used this CQ1 simulant from China. They had 4 treatments, so an untreated control, and then these 3 different biofertilizers, larger pots, 6 seeds per pot, thinning to 2 seedlings, and we get some growth here. We have the different fresh weight above and below ground. And so yeah, they actually saw a significant increase in available nutrients, so NPK and organic matter, when you added those microbial fertilizers, unsurprisingly. So yeah, nice little study of microbes. And enhanced lettuce growth and biomass accumulation resulted. So another microbe study, I think, again, showing a different aspect of using microbes in regolith. This is called a novel process to grow edible microalgae on Mars by exploiting in situ available resources, experimental investigation. So they actually grew spirulina algae as a potential food source for a potential Martian crew. So the idea is, you might be using microbes to help support plant growth, but you might just cut out the plant sort of end state altogether and just focus on microbes like they've done here. And that might be an important channel. And so they grew these. They use this thing they called Martian medium, which is a mixture of Mars regolith leachate and asteroid urine simulant. Turns out in space you can simulate anything. And they got biomass productivity that actually exceeded optimal growth medium in Earth conditions. And so here's just a diagram of sort of the closed system that this theoretically would go into. And here's their little clinostat. And these are the results. And so the ZM is Zarok's medium. That's like the ideal medium. And so they did samples with just air or ones with like high CO2. And we can see that the M is the Martian medium. We see that both in the air and this high CO2, those ones did better than just the default. They're not as good as the Zirkov medium or Zarok's medium and then high CO2. So, and then they also like evaluate sort of nutritional needs for a crew member and how this might accomplish that. So kind of interesting. And of course, you can't end without a fun little schematic of how this might look, years in the future on Mars. Definitely a little simplified, but a fun image. All right, let's shift over to fungal studies, notable fungal studies. There's a 2026 paper, selection of beneficial fungi for plants with the potential to metabolize lunar and Martian regolith. So fungi would be really beneficial for regolith-based agriculture because it's good at biomineralization, nutrient mobilization, and bioremediation. And so they looked at different groups. They found promising groups, different genre, as well as even the whole phylum, uh, Glomeromycota. Those are also known as the arbuscular mycorrhizal fungi, or AMF. And they even looked at fungal strains from the International Space Station that might be pre-adapted for off-world stresses. Some fungi are very good at coping with desiccation and abrasive mineral surfaces, which would make them particularly useful on regolith, which is desiccant and abrasive. And a particular usefulness they found in fungi is iron actually extraction and biomineralization. Though there are gaps in the field, you need better growth studies, more controlled studies, standardization, and actually look at biosafety, because some of the promising groups, promising genera, also have pathogenic species. You don't want to introduce pathogens when you don't have to. And you also need to improve inoculation systems and closed systems. All right, so some useful tables here, the different fungi they looked at and their different mechanisms, even the fungi that they found on or looked through on the ISS. And fun little picture here of possible pathways for fungal metabolization. All right, another study here was in 2026 as well, bioremediation of lunar regolith simulant through mycorrhizal fungi and plant symbiosis enabled chickpea to seed. So they actually planted chickpeas in different mixes of lunar regolith simulant and vermicompost for anywhere from 100% simulant to 25%, with or without inoculation of those AMF, those mycorrhizal fungus. And here's a little picture of there. They looked at different pH and it did shift. And why they chose chickpea, it's a legume crop, it has high protein, etc. So it's fairly stress resistant, so it would be a good use. And they found that, while plants grown in nothing but regolith did die, the ones with the AMF inoculation survived on average 2 weeks longer. So it improved. And the ones that didn't die, the ones that were on lower regolith percentages, there are some that with the fungi even developed flowers and seeds. So definitely useful. All right. Here's some of the data here if you want to go through it, the different metrics they did across groups. All right. Notable animal studies, because there's been actually a few animal studies. And this is my favorite name of any paper I've ever read. Turning earthworms into moonworms. And so this was a 2023 paper. They investigated the effects of the Lunar Regolith simulant, so the LHS1, at different concentrations in cow manure mixtures on the survival and fitness of these Acinia fetida. These are earthworms. You've got an example here, some of their cocoons, and then the simulant from Exolith. Well, it's now Space Resource Technologies. They did concentrations from anywhere from 0 to 100% regolith, and they had 14 to 60-day experiments. There was increased mortality in the 100% regolith, but most of the population survived overall. And that actually, the earthworm could potentially colonize lunar regolith in controlled environments, especially when you add the manure, you remediate the regolith with organic matter. Another animal study in 2025, improving lunar soil simulant for plant cultivation, earthworm-mediated organic waste integration in plant microbe interactions. So pretty similar. Earthworms were introduced into a wheat cultivation system with lunar regolith simulant and then solid waste, reed feces of some kind, or composting. And so there's a positive control, vermiculite, a pure regolith negative control, and then lunar regolith with solid waste, and then different levels of earthworms. They found improvements in compaction, salinization, microbial community shifts, and wheat growth parameters. And the solid waste and the similar lunar regolith were actually complementary. So, for example, one had high salt, one had low salt, so kind of balanced out. Or one was rich in organics, one was poor in organics, so they balanced out. So that was a good implementation. Bacteria genera were enriched. Some fungi genera were enriched, and the ones that were suppressed were probably pathogens, so that's even better. And so these results pretty useful for potential space missions, but actually this, and it's important to note with these regolith studies, it's not just for space. Actually, what we learn about dealing with regolith for a potential thing in space could actually help here on Earth. So we have dry, compact, low nutrient soil in need of bioremediation here on Earth. That methods that work in regolith could work here on those soils today. All right, so some bigger pictures here, the different measurements, the little view of how it all kind of work together. And then we'll kind of close out here with, we've focused on lunar regolith, but of course there's other types of regolith. A lot of times these reviews look at lunar and Martian together because those are the two main targets. And so here are just a smattering of papers on Martian regolith. So we have farming on Mars, where they actually had these nutrients contents. They made this briny water simulant and treated it with, I think, bacteria, and then they used it as a biofertilizer. They had this like system where they could sort of also filter the water through these various substrates, and then they could look at comparing, garden soil and fresh water, the regolith simulant soil and fresh water, and then like the filtered and biodesalinated water. Yeah, they desalinated with microbes here, and they looked at the different treated and untreated. There was the molecular study of Italian ryegrass grown on Mars regolith simulant, compared to Earth simulant. They did microscopy, proteomics, qPCR. They were able to get ryegrass to grow in Martian simulant, but there was stress issues, as you would suspect. And then there's a soil fertility interactions paper here, where they actually inoculated sweet clover with nitrogen fixing bacteria and potting soil and regolith. And so the inoculated ones in the potting mix obviously did better, but even the ones in regolith, they did a lot poorer on average than the potting mix. But you can see here there's definitely a pretty big difference between the inoculated and control groups in regolith. So adding that bacteria improved the plant outcomes. And I want to close with a fascinating kind of regolith that you might not have even realized is a real thing, is asteroid regolith. Actually, this study was done in 2022. Actually, I know two of the three authors here at my university actually ran this. And so they looked at the potential for primordial CI carbonaceous asteroid regolith for use in space missions. And they actually grew selected crops in increasing mixtures of the simulant and peat moss. So peat moss is acidic, so cuts out the alkaline nature of regolith. They did ratios of peat moss anywhere from 100% peat moss as sort of the positive control to 100% simulant, negative control, different ratios. They actually even did 3 crops: lettuce, radishes, peppers. They'd also ran gravity experiments. And they did find growth with their plants, but the simulant was prone to compaction, crusting, leading to drought stress on crops. And here's like the results here. You can see there's not even a bar for the 100% regolith. I think because they couldn't get anything to grow on 100% regolith, but if they mix it with a certain amount of peat moss, they could still get growth. So fascinating study about asteroid regolith. All right, with that is an overview of lunar regolith, and in fact other kinds of regolith for growing plants in. Here are all the references, and they should be found in the description of this video. Thanks for coming along with this wild, spacey ride with me. Definitely leave a comment, like this video, tell me what you liked about this, and I'd love to know what topic I should cover next. With that, Mark Williamson, thanks for joining me. I will see you next time.